Good morning, everyone. How are
Good
you
morning,
doing?
ma.
Morning day. Oluchi, how are you?
I'm fine, ma.
Good. How is your family?
Yeah, fine, ma. Everybody's fine. How are
All
you, ma?
right. Yeah, thank you all early attendees. Anoluwapo, Atanda Aishat, Benedicta Adewale,
Good
Divine
morning, ma.
Favor. Yes, Gbadamosi, Boluwatife, Oluwashimilore, Oshitono. Yes, Raji Ayomide, Said Zainab. TS, I don't know what is TS. So another Zainab, Samuel Akinwale, Stephanie. So welcome again this morning. So while waiting for the remaining 2 minutes or so, I would like one person to unmute and summarize what you learned on Friday for me. If you have 230801137, please kindly add your name. Otherwise, I won't be able to take your attendance. So I'm looking for someone to tell me what we learned last week, Friday. Benedicta, do you want to say something? Who wants to say something? So now I will call on someone to summarize. Okay. Said Zainab Bishola, cell biology. Can you just tell me one thing you have learned last week? You can unmute and tell me that one thing that you learned last week.
Good morning, ma.
Good morning, dear. How are you?
I'm fine, ma.
Thank
My
you.
network was not stable, so I was not able to join the class. That's why I came early this morning.
Okay, so since you didn't join the class, I sent the video recording, I sent the live link. You didn't bother to go through it.
It's ma.
You are waiting for exam to come before you start to read. This is biology and not mathematics or statistics where you can apply formula. Here you have to really read and understand. And for you to read and understand, you must have gone through your note at least 3 times before the exams. So Alade Princess Fatia, biochemistry. Thank you. Just tell me 1 or 2 things that you learned last week.
Good morning, ma.
Good morning, dear.
I wasn't able to continue the class to the end, but I learned that microorganisms have different roles in our environment.
Okay.
They have different roles in the air, in the water, and on land. So,
Okay, thank you. At least you picked something. Is there any other person that wants to unmute and tell me what he or she learned last week?
Yes, ma.
Okay, is that Oluchi?
Yes, ma.
All right.
So I learned about microorganisms in relation to the environment. For example, bacteria, they play a role in nitrogen fixation and decomposition. And then for fungi, they are decomposers. For virus, they kill bacteria to reduce population, the bacteria population. For protozoans, they engulf things that are not meant to be there. Like, they just engulf things.
They
Then
engulf even
algae
bacteria. They also help in controlling the
population.
microbial population in the environment too.
Okay.
Thank you very much, those who have spoken. I appreciate your your contribution. I want to encourage everybody. See, biological courses are not the courses that you want to put on one side. I hope you will agree with me. Most of you, biochemistry, marine, we're all in this business of biology together. We know that biological courses are voluminous. So they are not the kind of courses that you want to leave and say, okay, 2 weeks to exam or 5 days to exam, I want to start to read. It will be too late by then. So we'll go into today's lecture. As usual, I want to go off my video so I can have a steady internet connectivity. You can do likewise so that your internet can be stable. Thank you. So today, we want to move to the last topic for this semester, microbial variation and heredity. Hopefully we'll finish today. If we're unable to finish today, then we'll conclude on Friday by the grace of God. The slides are already prepared. That is why it is easy for us to move fast this way. So please, I want you to cooperate, mute your mic so that we can have a smooth lecture this morning. So microbial variation and heredity. At the end of the lecture, you should be able to explain the concept of microbial variation and heredity. Let me make it clear that this is basic microbiology. It's like introduction. So everything I'm teaching you, we are teaching you in this MIC 211 is introduction, which those going higher with microbiology will now pick as courses. For example, this topic is a whole course, it's genetics, MIC 401. So, but you are just learning like the food we did, role of microorganisms in food is a whole course. Food microbiology, industrial microbiology is another whole course. So we're just giving you the tip of the iceberg. So not going too deep. So whatever I want you to know, just know that your questions are coming from your slides and it is a CBT question. So you should be able to know the concept of microbial variation and heredity to be able to give some concept, some definition to show that, okay, you have and understand the basis so that when you go higher, for those in biochemistry, microbiology, and cell biology, definitely you will go higher. You should be able to identify and describe different types of genetic and phenotypic variations in microbes. You should be able to discuss the mechanisms of genetic variation, including what is mutation, recombination, and horizontal gene transfer. And you should be able to illustrate how microbial heredity contributes to genetic stability and diversity. You should be able to apply knowledge of microbial variation in real-world contexts, such as in antibiotic resistance and biotechnology, which we have spoken about in the past. Introduction. Microbial variation and heredity refers to the genetic and phenotypic differences between microorganisms. And this variation or differences is what normally contribute to adaptability of a microorganism or any organism at all, including human being. I'll be using human being as example for you to understand, so you'll be able to apply it to microorganisms. So the variation or differences that we are talking about microbial variation, that is differences between organisms. Then we can have genotypic differences or phenotypic differences. This variation is what allows organisms, like microorganisms, to adapt or to survive and to evolve. So our understanding of microbial heredity is very necessary in fields like medicine. If you don't understand what heredity or genotype is, you will not be able to study medicine, biotechnology, or microbiology very well. So, sorry, my computer is hanging, so I have to end the show and All right, so definition of terms. So let's look at some definitions that we need here. So what is heredity? Heredity simply means biological inheritance. Heredity is the likeness of the characteristics of a progeny, that is an offspring and the parents, in human being. In human being, it is that what makes your offspring to look like you. The same thing applies to microorganisms. It is those characters that a particular microorganisms have inherited from their parents that make them look alike. So it is the process by which exact transmission of genetic material takes place from parents to their progenies, that is to their children or to their offspring, through sexual or asexual reproduction. So there is resemblance among organisms. Why is it that if I see a plate of Bacillus thuringiensis, I can say, okay, this is Bacillus thuringiensis. I can say this is Bacillus thuringiensis. If I see a plate of Streptomyces, I can say this is a plate of Streptomyces. It's because they resemble each other. But if anything goes wrong, I will know that something has gone wrong with this organism because it is not looking like what it's supposed to look like. So that leads to variation. Variation is the difference between progeny and parents. That is, variation refers to a change in an organism compared to their parents. We have inheritable variation. That is, variation that can be inherited by the children. Let me use a human being as an example. Maybe there's a particular disease or there's a particular, maybe the family used to be fair in complexion or they used to be tall or something. Those are physical stuff. But anything that has to do with genetic, maybe like a particular disease, and the offspring can inherit it. And something goes wrong because of situation. This people have started. Please mute. So that variation is whatever goes wrong with a particular organism that has to do with the gene can be passed on to the parents. If, for example, mutations, mutation takes place, if, for example, mutation takes place and is passed to the generation to come, and that mutation, because it is genetic, it will be passed, it will be transferred to the progeny, to the offspring. That is why it is inheritable. You can inherit it. So inheritable variation is brought about by changes in genetic substances. That is when things like mutation, loss of acquisition of new genetic elements happens within an environment. You also have heritable variation. Sometimes people mistake heritable variation with inheritable variation. They look similar, but they are a bit different. Inheritable variation has to do with something that goes wrong with the gene, mutation, loss of acquisition. Heritable variation can occur whether it is genetic or not. Non-heritable variation is the changes of variation that is brought about by environmental changes. So this kind of change is not permanent because they are basically phenotypic. For example, maybe the family are normally tall, but they give birth to an offspring and something happened, and that offspring have to find him or herself with the grandparent in one village. Maybe the child became malnourished, very sick, and they didn't take, and then the child grows very short or stunted. That is a physical stuff. It is not a genetic stuff or change or variation. It is physical. It has to do with the environment that the child finds himself. So what happened is, because it is physical, it is phenotypic. So it can be reversed. It is not permanent. But if the change or variation has to do with gene, the genetic, it's going to be a permanent change. It will be carried out from the offspring of that person on and on. But for the non-heritable, it's come about by the environment. So for microorganisms, maybe a microorganism supposed to be cream in color, but you now place it on a very high temperature, it can change to red. But if you pick that same red organism and you put it in the media and put it in an appropriate incubation, it will go back to its cream color. So that is what non-heritable variation means. Then we have phenotype. Phenotype is the characteristics expressed by a cell at a given time. Take, for example, human being. You are black, you are only expressing your black forever. You are short, you have red eye or brown eye or cat eye, whatever. Those are physical stuff. Your hair is straight or your hair is curly. Those are the physical characteristics. So also it is with microorganisms. Some have round colony, some colonies are wrinkled, some colonies are raised, some are flat. Those are physical things. That is why they are called phenotypic characteristics. So when you have phenotypic changes, that is phenotypic variations, under uniform conditions, bacteria will possess constant characteristics. This makes bacterial identification and classification possible. So, but when you isolate pure culture and you store them in the lab under the same proper condition, they remain the same. But if you change the conditions and you want to study these organisms, they may change. The appearance may change, like I explained earlier. The condition can make appearance to change. You come to the Western countries, because of the weather, your body is fresh. You go back to where you have the tropic, then you begin to turn black. That does not mean that you cannot become clean again if the temperature becomes okay. That is what phenotypic changes or variations has to be. Genetics is the science of heredity and variability of characteristics of an organism. The study of heredity and variation is what is called genetics. There's another word you must know, the genome. The genome refers to the entire gene possessed by the cell. That is, microbial genome encompasses all chromosomal and extrachromosomal genetic material. The human being have their gene, they have their genome, and most of the time the genome have numbers, which everybody knows. Genotype is the genetic constitution of a cell. That is what is specific about your DNA. So the DNA for a particular individual or a particular organism is what is called genotype. Some people in human beings are AA, some are AS and all that. So in microorganisms, they have their genotype. They have their own type of DNA also. Then genomics is the study of genomes of an entity, of an organism. Don't confuse genetics with genomics. So microbial variation. Microbial variation refers to the differences in genetic material or observable characters among microbial populations. And this variation can be categorized into genetic variation, which I've explained during our definition of concepts. Genetic variations are inheritable and result from changes in the microbial genome. And this could be as a result of mutation. Mutation is the sudden and inheritable changes in the nucleotide sequence. That is the protein that is in the DNA. It may also be as a result of recombination, that is the exchange of genetic material between two different DNA molecules. It could also be as a result of horizontal gene transfer, that is the transfer of genetic material between bacteria. And this could be as a result of transformation, transduction, or conjugation, which we will look at briefly in the course of this lecture. So what are the mechanisms of genetic variation? Mutation. Mutations can be spontaneous, that is, it can just happen. That is what happened to cancer cells also in humans. It started one day, so anything could have happened. Nobody knows the actual causes of cancer, for example, but they do know that there are some disposing factors. So if because of one reason or the other, the liver or the lung or any part of the body, the cells begin to mutate, it will be different from what it's supposed to be. So spontaneously, cancer or any type of disease can just develop. And if it has to do with gene, it can be transferred to that person's children later in life. So the same thing with organisms. Mutation can be spontaneous, it can just happen, or it can be induced by external factors, such as chemical or radiation. If, for example, you go to an X-ray, they will tell you when the light is red, don't enter the X-ray room because of radiation. But if somebody carelessly enters, the person might have cancer. So also organisms, there are some chemicals that can mutate or cause mutation with microbial cells. So it makes them to change from what they are supposed to be. And we have different types of mutation. We have the point mutation, which is as a result of substitution, insertion, or deletion of single nucleotide of DNA. We can have frame shift mutation, which is the addition or deletion, causing a shift in the reading frame of the nucleotide. We have the silent missense and nonsense mutations. And this kind of mutation affect protein synthesis. So when protein synthesis is affected, definitely the DNA nucleotide will be affected. Another mechanisms for which we can have genetic variation is by recombination. Genetic recombination involves the rearrangement of genetic material, contributing to diversity. So when genetic material is kept rearranging itself, you keep having diversity of different organisms. That is why we'll have different species and different strains of organisms. Well, okay, Bacillus subtilis, strain so. You see that the strain that is evolving in Africa is different from the strain that is evolving in America. Is because of rearrangement of genetic material that contribute to diversity. And this can occur through one, homologous recombination, that is the exchange between similar sequences of DNA. It can be through site-specific recombination involving specific sequences and enzymes. And it can also be through horizontal gene transfer, which involve transformation, transduction, and conjugation, which I will explain. So, microbial heredity involves the transmission of genetic information and parent cells to their offspring. So this is the structure of DNA, what it looks like. Last week we talked about their DNA being simple. This is bacterial DNA. It's just a single strand DNA that is circular. But microorganisms can also have extra chromosomal DNA, which is called plasmid. And most of the time, plasmids are responsible for antimicrobial resistance in microorganisms. So bacterial chromosome, usually circular and lacks a nuclear membrane, as you can see. And they also have plasmids, which is an extra chromosomal DNA. It's not all organisms that have plasmids. So those who have this DNA carrying non-essential genes, often for antibiotic resistance. Then gene expression and regulation. We have operon model, example, the lac operon in E. coli. This is the lac operon structures here. And you can see that it's the structure have a promoter, an operator, and a repressor. So it has enzyme, beta galactosidase, that I can use to break down galactose. So, but that is too much for your level. Just look at, this is the picture of what lac operon structure look like. And it is one of the structure that is used to express and regulate gene in bacteria. You also have regulatory protein, activators, and repressors controlling gene expression, that you can see on the lac operon. We have the epigenetic modifications, DNA methylation, influencing gene activity in bacteria or in microorganisms. So let's look at phenotypic. We've just talked about the genotypic variations and what can cause them or what can be responsible. So for the phenotypic variation, we said phenotypic variation occur due to environmental influences and may not be heritable. This includes changes in metabolic activities, antibiotic resistance, biofilm formations. These are phenotypic and they can be reversed. Morphological changes is one major phenotypic variation. Morphology, the appearance of an organism. Morphology is the branch of biological science that deals with the study of the structure and forms of living organisms. So the structure of an organism can change due to phenotypic variation. For example, when a bacterium is inoculated onto a fresh medium, it undergoes different phases of growth. So the cell may appear large at the end of the lag phase, which is the actively growing phase. But as it proceeds through the log phase or exponential phase, you will do that in MIC, second semester MIC. And it approaches the stationary phase, the cell will appear small, but though they are small in size, the sizes will be uniform because of the cell divisions. So different phases of growth of microorganisms can be responsible for their structure or their morphological changes. So the organism that is growing at the lag phase may appear large, and towards the stationary phase may appear small, but it's still the same organism. It is this phase of growth that is causing that morphological changes or variation. Another kind of change that can occur in microorganisms is cultural changes or variation. Cultural changes, culture, that is the culture of the organism on plates. So one of the changes that can occur with the culture of a plate is pigmentation. Pigmentation means coloration. So very important, this pigmentation is very important in cultural characteristics of organisms, of microorganisms. We use pigmentation to identify organisms. So when anything gets wrong, if I pick my Streptomyces species and it turns green, I know something is wrong. If it is not contaminated, the color should not change from cream color. So pigments production by microorganisms is usually affected by incubation temperature. For example, an organism called Serratia mycescens produces distinct brick red pigments when incubated at room temperature. But if you put Serratia at 37 degrees centigrade, it will lose its pigmentation. Pigmentation is completely absent. So for somebody who is not used to the organism, you will argue that this is not Serratia. But it is the temperature that has caused the organism to lose the pigmentation. Another cultural change or variation that may occur in microorganisms is shape and structure variation. Colonial changes, the colony of the organism, changes or variation can occur in microorganisms. For example, the ability to produce capsules can result in changes in colonial form of some bacteria. The synthesis of polysaccharide capsules by bacteria and producing large mucoid colony in some instances occurs only when the media contains disaccharide sucrose. So if I don't want my organism to produce mucoid colonies, I will not add disaccharide sucrose. So it will change the structure. Another type of media, the organism may appear smaller because no capsule is produced. So if I don't feed it with what it's supposed to produce capsule with, which will help it to grow well, it will not produce the capsule. It is not all organisms that produce capsule, but one major organism that produces capsules is Mycobacterium tuberculosis, the organism that causes tuberculosis. And the reason is that capsule is like a protection to the organism. Before the antibiotic can destroy that organism, it must first destroy the capsule. That is why it is difficult to treat Mycobacterium infection. Smooth colony may also change to rough colony when the environment becomes unfavorable, when there's no enough nutrient. A colony of microorganisms that's supposed to be usually smooth can become wrinkled. Then we can also have physiological and biochemical variations. The resistance of microorganisms to harmful chemical and physical agents varies, for example, with age. So physiological changes can be affected by age, which is also the same thing with human beings. So young cells are more susceptible to disinfectants. So when we have young cells of microorganisms, the new cells, they are easily destroyed by disinfectant. But the older cells of microorganisms are more resistant because they are already used to this disinfectant. So it doesn't affect them any longer. Resistance are taking place. Also, the type of enzyme a particular species of microorganism will produce depend on the kind of environment or the substrate that it is growing in. For an organism to release an enzyme, that means there's something that is stimulating that enzyme. That is the substrate or whatever food or media you feed the organism in. For example, induced enzyme formation occurs only when the specific inducer is present in the medium. The medium, that is the food or substrate you are giving the organism. Look at E. coli. E. coli can release beta-galactosidase enzyme, but it can only release this enzyme when it encounters beta-galactose. That is, if you put beta-galactose sugars in the media, the E. coli is going to release beta-galactosidase to break it so that it can be able to metabolize it. Beta-galactosidase is that enzyme that splits or breaks down galactose sugar. In terms of genotypic and phenotypic, or in terms of genotype and phenotype, this means that the ability to produce the enzyme is inherited in its genotype. That is, if that organism does not have the genotype to produce this enzyme, it will not produce it, despite the fact that you may put galactose. So if I put galactose and the organism did not release beta-galactosidase, that means it does not have the genotype. It is not in its genes to be able to do so. But whether or not the enzyme is produced, phenotypic expression of this character is determined by specific circumstances in its environment. Genotypic changes, we have explained all that. So, but we know that mutation is stable. When mutation occurs, because it is a genetic problem, that change is stable. There will be a change in the gene of that bacteria, and the act of mutating is called mutation. So let me give you an example. If a group of microorganisms, of a colony of microorganisms is attacked by bacteriophage, you know that bacteriophage is phage, viruses. And you know phages usually attack bacteria to destroy them. But after this attack, there are some bacteria that will survive. Once they survive, they become very resistant to phage. Once they survive, they become resistant to phage. And they carry this resistance to their descendants, that is their offspring, so that their offspring can no longer be affected by phage. What is the difference, or what are the differences between phenotypic and genotypic changes? Phenotypic change involves most of the cells in the culture, while genotypic change can take place only at a single cell level. So even if you have many cells in a plate or in a medium, in a flask, when genotype wants to change, it will start with one cell. It may affect one or two cells, but not all. But phenotype will affect the whole cell because it is the same condition that is affecting them, the same environmental condition. Phenotypic change is less stable than a genotypic change. When environmental conditions return to normal, a return to the original phenotype can occur. Phenotypic change is not inherited, while genotypic change can be inherited. Let's look at the gene and the DNA. The gene is the arrangement of nucleotide pairs of DNA, which encodes for a particular polypeptide chain. That is, genes are made up of DNA. The gene is found in the chromosome. DNA is deoxyribonucleic acid, and it is the molecule that accommodates the genetic code of organisms. That is the structure that conveys the details for which human beings or microorganisms are made up. That is what contains the detail of your attributes. So it determines the attribute of the cell of a microorganism. So DNA is the vital material in the transmission of genetic information in organisms. The chromosome is a single large circular DNA in bacteria. I've showed you that one before. In some viruses, the chromosome is composed of RNA, that is ribonucleic acid, and they cannot have the two. It's either they have DNA or RNA for viruses. The molecular weight of chromosomes in bacteria, for example, E. coli, is 2.5 times 10 raised to power 9 Daltons. And it contains about 3,800 genes. We can study this. When we isolate organisms, we do carry out molecular studies to know what is the number of their gene. So if anything is wrong, you can know. Even in human being, you born a newborn baby and they carry out all these tests and they saw that something has deleted. It is not up to what it's supposed to be. They can easily fish out where is something wrong. So in small bacteriophage, it is like 1.7 times 10 raised to power 6 Daltons with 7 genes only for the bacteriophage. Bacteria are almost always haploid, that is, they are haploid. Their chromosomes all have the same genetic structure, or that they are unpaired, while for eukaryotic cell, they are diploid. So what are the differences between the DNA and the gene? Genes are DNA stretches, which are encoded for different proteins. DNA is a chemical that stores the genetic information of an organism. The gene regulates the traits of an organism. The DNA regulates the gene regulation. The gene is the small stretch of DNA. The DNA is the long chain of polynucleotide. The genes are made up of either DNA or RNA. The DNA is basically made up of nucleotides, which are protein sequences. A gene is located on a chromosome. DNA is located within the nucleus of the cell. Genes are coded with heredity information. DNA encodes genetic instructions. And this is the diagram. This is the relationship between the chromosome, the gene, and the DNA. This is the DNA. You can see this enlargement here, where you have guanine, thymine, cysteine, adenine. These are the nucleotides. Always for here. So you have DNA, and it is enclosed within the chromosome. So the whole thing is now what is called the gene. So let's look at how genetic materials are transferred in prokaryotes like microorganisms. Genetic recombination is the formation of new recombination of genes. That is, recombination is the formation of new genotype through reassortment of genes of two different cells. It occurs between two different chromosomes having identical genes at the corresponding sites, that is the homologous chromosomes. But because bacteria are free-living haploid organisms and they are not differentiated into gametic or somatic cells because they are prokaryotes, each cell has the potential to behave as a gamete. So bacteria can just keep dividing and they will keep multiplying. For a bacteria that have maybe 6 or 8 hour ability to multiply, they double every 6 hours or double every 8 hours or double every 12 or 24 hours. Different microorganisms have different doubling period. They are the same cell because they are not differentiated into sexes. Though some of them display sexuality, but the cells have just to differentiate themselves into donors and acceptors, while the donors are the males, the females are the recipients. So in bacteria, only a fraction of the genetic constitution of the donor cell can be transferred to the recipient cell. The recipient cell is a merozoite, a zygote that is completely diploid. So we can have different methods for different ways by which microorganisms transfer their genetic material from one organism to another to multiply. And I mentioned transformation, conjugation, and transduction. So let's look at transformation. Transformation is the genetic alteration of a cell resulting from the direct uptake or incorporation and expression of exogenous genetic material. Exogenous, that is, the organism is picking up this DNA material from the environment outside itself. Maybe it's in a flask with other organisms, or in the soil, or in the water. But around, you have different DNA. And somehow, this organism picks up an external, what we call exogenous DNA from its surrounding, and then replace it, maybe delete the one it has and replace it with the exogenous, or add it. That's what we call addition or deletion. And this occurs in the environment through its cell membrane. So transformation can occur naturally in bacteria, but it can also be done artificially. That is why we can have genetic engineering now. If a cell in human being has a problem, they can delete where the problem is, the gene that has a problem, and replace it with another one. You've heard of a kidney transplant, the liver transplant. That is where they got all those knowledge from. So bacterial transformation can be referred to as a stable genetic change brought about by the uptake of naked DNA in the environment. But you know what? Before a bacteria can successfully pick up an exogenous DNA into itself, it has to be competent. If that bacteria is not competent to do so, it will not be able to do so successfully. You have seen that some kidney transplant can fail, liver transplant can fail. Even blood donation, some people take blood, they react. So if the organism is not competent, it will not be able to do so. So competence refers to the state of being able to take up exogenous DNA from the environment by bacteria. So some bacteria we acquire and then subsequently transmit a character of the related strain when grown in the presence of dead cell. Culture filtrate or cell extract. So when they acquire this DNA, whenever you take them to, they will grow and they will acquire the DNA and they will transfer it to their progeny or their offspring. That is where different varieties of organisms start developing. So this is the diagram that can show you what is happening in the environment. We are just using flask and all this to demonstrate. So if you have organisms in this flask, it could be water, it could be ocean, it could be fermentation taking place, any environment. Look at DNA fragments of different bacteria in this environment. And then you have these bacteria outside and they are in the same environment. These bacteria have its own nucleus, which I said is always circular and single-stranded. So if they find themselves in the same environment, you can see that through the cell membrane, the DNA fragment that is in the environment that is free enter the cell of the bacteria because this bacteria is competent, it's capable of allowing it. So two things can either happen. If this organism is not competent, new DNA will be degraded. So it will not be able to absorb it into itself. So we call this unsuccessful transformation. But if the DNA is integrated successfully, the bacteria is competent, you have a stable transformation. You can see that the DNA that we color in red for you to know what is happening is successfully embedded in the bacteria normal DNA. So it is a successful transformation. And if it is an organism that have plasmid, the same thing. If you have plasmid in the environment, the plasmid DNA can be incorporated, can be taken up by a bacteria in the environment successfully. Another method by which microorganisms acquire DNA from their environment is through conjugation. Conjugation is the process by which one bacterium transfers genetic material to another through direct contact, that is cell to cell. During this conjugation, one bacterium will serve as the donor, that is the male. It will serve as the donor of the genetic material, and the other will serve as the recipient, F, maybe like the female. So the donor bacterium will carry the DNA sequence called the fertility factor or F factor. The F factor allows the donor bacterium to produce a thin tube-like structure called pilus, which the donor uses to contact the recipient. The pilus then draws the two bacteria together. The donor bacterium transfers genetic material to the recipient bacterium. So what I'm trying to explain is in the next slide diagram. The evidence for conjugation was described by a scientist called Lideberg and Tatum in 1946. So this is the structure. This is the mechanisms of bacterial conjugation. This is representing the F fertility. This is the donor. So we are using this red to show what the portion of the DNA that will be transferred, the genetic material that will be transferred. So you can see that the one representing the donor produces a pilus. Then it will come to the recipient. Pilus connects the cell to the recipient. Then the donor DNA replicates by rolling circle method and transfer part of its genetic material to the recipient. You can see the process of transfer here. And then the genetic material that is being donated is attached to the genetic material of the recipient. And this has been successfully transferred. You can see that what we painted red is now here. So this one now, by the time this organism multiplies or grow, the offspring of this one will have the DNA of the donor, and it is transferred from generation to generation of that offspring, except something goes wrong, like mutation. The other method is transduction. Transduction is a process of genetic recombination in bacteria, where which genes from a host cell, that is a bacterial cell, a bacterium, are incorporated into the genome of a bacterial virus, a bacteriophage. And this is then carried to another host cell when the bacteriophage initiates another cycle. There are two types of transductions, generalized and specialized transduction. But the generalized transduction, it occurs during the lytic cycle of virulent and temperate phages. Specialized or restricted transduction, the transduction or the transducing particle carries only specific portion of the bacterial genome. So this is what I'm trying to explain. Transduction of bacterial gene by viruses. This is a bacteriophage. Bacteriophage is a combination of bacteria and virus. This is the head of the virus called phage. It has bacteria. So now in the environment, there's an innocent bacteria in the environment. This is the chromosome containing the genetic and DNA particle of the bacteria. But this bacteria, the bacteriophage, the viral phage, is coming from outside and perches itself on the bacteria. So the phage injects DNA through the cell membrane of the bacteria. And the phage has an enzyme. It will degrade the DNA of the host, that is the bacteria. You can see that that round chromosome has been destroyed. It has been cut into pieces by the enzyme of the phage, of the bacteriophage. So what will now happen? There will be cell synthesis, new phages with them. It's like it injected its own DNA and then started developing inside the cell of the bacteria in the environment. So cell synthesis, new phages now occur. So you can see that it is giving rise to different phages inside the bacteria. So the phages with donor DNA transducing the phages. You can see multiple phage now inside the bacteria. So the recipient cell transducing phage injects the donor cell, and then the cell is transduced. This is the new phage, the one from here, the one it gave birth to, that the cell synthesized in number 3, is now standing alone here, and the process will continue. Then you keep having transduced cells that will be carried from generation to generation of this specific organism. That takes us to Mendel's law of inheritance. So, but before we move on to Mendel's law of inheritance, I will not ask you to unmute because of some of you that are always disturbing. Some people are not, they are just marking register. They are not here to listen to lectures. But if you have any question, type it. I will check it later. So we move into Mendel's law of inheritance. Mendel's laws of inheritance. This is just to remind you of what you learned in secondary school, because we all did Mendel's law in biology. Just for you to know that this law is still useful for biotechnology and for gene transfer, just for you to understand what is happening in genomics. So the name of the scientist, the father of genetics, is called Gregor Mendel. Gregor Mendel is the father of genetics. He discovered 3 laws of inheritance. He discovered the law of dominance. That's the first law. The law of segregation. Then the law of independent assortment. We'll go back to those laws one by one. Mendel described the basic pattern of inheritance, even before the mechanism of inheritance, that is, before gene was discovered. He did not know about gene. There was nothing like genome. Nobody knows it, but he described what is happening then. And Mendel he conducted many experiments using pea plants, Pisum sativum, between 1856 and 1863. He studied the result of the experiments and deducted many observations. He made his observation known. So the law of inheritance of Mendel's came to existence. It is important that we understand the experiment performed by Mendel before learning the law of inheritance. So let's look at some of those experiments. Mendel experiments on pea plants. Pea plants, that's pea is like beans. Pea plant was selected by Mendel. Why? One, the pea plants easily increase in size. So it is easy to observe, it is easy to study. The pea plant has distinct characteristics that are easy to conceive. So it makes it easy for Mendel to study the characteristics of the pea plants. Because this plant is available annually, it is not a plant that you say, oh, you cannot see it during the rainy season, or it can only be available during the dry season. It chose to use a plant that is available all year round. Many of its generation can be studied within a short period of time. And it chose pea plants because pea plants can pollinate itself naturally and can also be cross-pollinated. So that there are those two advantages of using pea plants. And he knows that whatever happened to a particular organism is most likely happening to other organisms with some little exceptions. So he listed some contracting characters which he studied. He studied the following. He studied the color of flower of the pea. He studied the height of the pea. He studied the seed color. He studied the shape. He studied the pod color of the pea and pod shape, and then the flower position of the pea. So during his studying, he discovered that the flower color can have dominant purple. The plant height dominantly is tall. The seed color can be yellow. The shape can be round or wrinkled. The pod color can be green or can be yellow. The pod color may be inflated or constricted or flat. The flower position may be axial or terminal. And look at the dominant trait he has listed, and then the recessive trait he has listed. So in following, you can use any of these traits to study Mendel-Gregor's experiment. So sometimes you can see capital PP, small pp, capital TT, small tt. When you say a capital P, it means dominant, the trait that is domineering that you can see. The recessive is that trait that is existing in that organism, but that you cannot see physically. So he uses a pair of letters to represent each of the traits. For example, a pair of letters T. Capital T is for dominant traits. Small t is for recessive. Recessive means it is there, but you cannot see it physically because the capital one is dominating. So the tall, let's say T is for tall, and the small t is for short. So this organism is appearing tall of the dominant trait of the capital T. Or YY. These are two capital Y for color yellow. So this organism, Olanike, you are disturbing. Olanike, mute. All right, so the YY, they are both capital. So that means that this organism is yellow, is completely yellow. If it has a recessive Y, that means it is dominating. The capital letter is dominating, but this organism, for example, has both capital Y, so it is a yellow color without any recessive yellow. And you see SS, small, two small s, so small. Short. So this organism is completely short because it has two small s. So he uses this alphabet to translate what he wants to let us know about the character of what he's studying. And it is very necessary for you to understand. I know you know it from your secondary school days. So a pair of letters represent genotypes for a particular trait. There are always two letters in the genotype. There are three possible types or genotype. TT, completely tall. Capital T, small t. Tall, appearing tall, but have a recessive shortness. Then TT, completely short. The genes that are passed from the parent to the offspring exist in pairs. These pairs are called alleles. Dominant characters are described using capital letters and recessive using small letters. When the two letters are the same, that is, if you have the same allele, capital T, or TT, small t, they are called homozygous. When both the alleles are different, they are called heterozygous, like capital T, small t. That is, this organism appears tall, but it has a recessive gene for shortness. It is called heterozygous. Hetero means other, also referred to as hybrid. When the plant appears tall, has the recessive gene, which might express itself in the future generation. So for example, this one with capital T and small t, the organism though appears tall because the tallness is more dominant. But don't forget that the recessive gene for shortness, the small t, is here. So the organism, maybe the second or third generation of this person or this organism will be short, because that short genes might later appear in one of the offspring later, even though the organism is appearing short. In human being, you now say that your mommy is tall, your daddy is tall. Where did this child get his shortness from? Is he a bastard? No, it must have been that one of the parents, forefathers, have the recessive gene. The same thing with people that are, what do you call them? They are thin. Where did they get this one from? It is only a gene that is coming to show up from the forefathers. So also it might happen with microorganisms or any other plant. So when the plant appears tall, has the recessive gene which might express itself in future generation. The way the plant is known as phenotype. How the trait physically show up in the organism, that is phenotype. It doesn't matter, it has the recessive gene, small t for shortness, but it is showing as a tall organism. AS have a sickle cell hidden. So if AS and AS come together, one day they will have a sickle cell. Even if they have AS, one of their children might have SS children later. So it happens with all organisms. So when the genetic makeup of the plant is called the genotype, it's called the genotype. The genetic makeup of the plant is called the genotype. Hence, the observable characteristics of the plant with TT, that is capital T, small t, genes appears tall, but has a recessive gene for shortness. So let's go to the first law. The first law, the law of dominance. All right, so the first law, the law of dominance. Law of dominance states that in a heterozygous condition, heterozygous, don't forget, heterozygous means it has a recessive gene. The allele whose characters are expressed over the other is called the dominant. So let's look at this experiment, this flower pod. Let's assume these are the parent generation. And one of the parents is completely red. The other parent is completely white. For their first generation, their first offspring, what will happen? The red parent is donating one of its gene, red, capital R. The white is donating a small r. So for the first generation, they have an heterozygous offspring that looks red, but have a recessive white gene embedded in its gene. That is what the law of dominance says. It states that in a heterozygous condition, the allele whose characters are expressed over the other one is called the dominant. So here now, for the first F generation, the red plant dominates the white because the plant appears red. But this plant that appears red has a recessive white gene in it. Okay, so we move on to what is called Punnett square. Sorry, I don't know what is happening to my, everything is mixing together, okay? So we have what is called Punnett square. Punnett square is named after Reginald Punnett. The Punnett square is a square diagram that is applied for the prediction of the genotype of a particular cross of breeding experiment, whether human being, plant, microorganism, it is the same thing. So you use the Punnett square to carry out your cross-breeding experiment. The square diagram is employed by biologists to determine the probability of an offspring having a particular genotype. Don't forget, cast your mind back to what we have done here, first generation. So we continue from here using the Punnett square. So if you now bring together that first generation, which has one tall and one recessive gene, and you crossbreed it, you are going to have another type of character showing up. Parents, that is P, with tall that have dominant tall, TT, and short one, TT, small t. Let's look at what happened. Split them, because we said that they always have two genes. It is pair. So this one, capital T, that is TT. Then the recessive one, C and T. So use the Punnett square to crossbreed. What will you have? Capital T and small t will give you one capital T, small capital T. This and this will give you one capital T, small capital T. This tall, that is capital T, and this one will also give you one capital T and small t. And then this and this will also give you capital T and small t. So here, with this one that we have done, what happened? Even though we have two short parents coming together, two parents, one that is completely tall and one that is completely short coming together, their children all appear tall, but all their children have the short recessive gene in them. The same thing, if you take a person with SS and you allow them to marry AA, they will never have SS children. All their children will be AS. This is exactly what is happening here. So you have 100% offspring that have or appear tall, but all of them have the recessive gene for shortness. I hope we understand. Why is this thing hanging? Okay, so that now takes us to the law of segregation. We have just completed the law of dominance, how characters dominate one another in an organism. So how does this law of segregation applies? How does this law of segregation now applies? The law of segregation. This law states that when two traits come together in one hybrid, remember, cast your mind back here so that you won't forget. You have 4 offspring, all with tall and T, small t, and 1 tall t. So we are bringing them here to the law of segregation. The law states that when 2 traits come together in one hybrid pair, this is an hybrid pair here, the tall t and the small t. We've taken one of them, they are both now coming together, 1 small t, 1 capital T coming together. The law of segregation states that when these two traits comes together in one hybrid pair, the 2 characters do not mix together. These tall and short do not mix together. They are only appearing tall, but these 2 genes are different. They do not mix together, and they are not dependent on one another, so you can separate them. Each gamete accepts one of the 2 alleles during meiosis of the chromosome. That is, each organism has 2 alleles, and a parent transfers just 1 allele to the offspring. It cannot transfer the 2. If the parent decides to transfer the gene for shortness during reproduction, that organism will be short, depending on what the other partner is also donating. This is one partner, this is one partner here, 1 here, 1 here. This is one, this is the other. So if this one is donating small short and this one is donating small short, that offspring will be completely short. So each gamete accepts one of the 2 alleles during meiosis of the chromosome. Each organism has 2 alleles, and a parent transfers just 1 allele to the offspring. The factors must separate themselves somewhere between the production of sex cells and fertilization. So from the P² above, it can be deduced that when you cross 2 hybrids, that is from our F1 generations, 3 of the 4 boxes will produce an organism with dominant trait TT. Look at it. He said you must segregate them. You have an organism with dominant tall, capital T, but with recessive gene for shortness, small t. He said segregate because they do not mix together. Because during reproduction, this organism can only release one of these characters to the partner. So separate them. You have 1 capital T, 1 small t. This is the partner. It also have a tall character, but with recessive gene for shortness. So separate them, capital T, small t. So what will happen? Crossbreed. This capital T with this capital T will give you capital T and capital T. That means the organism is completely tall without any recessive gene for shortness. If you crossbreed this capital T with this small t, the organism have 1 capital T, 1 small t. That means the organism appears tall, but it has a recessive gene for shortness. If you take this small t of this offspring and this tall, you have a tall organism with a recessive shortness. But if you take the shortness of this partner and the shortness of this partner, they come together, they have a completely short offspring. So you can have a family, they have 4 children, 3 are very tall and 1 is very short. You'll be wondering, why is this one different from the other? This is what is happening in the gene. So for this one now, the organism, I want somebody to unmute. Tell me, how many of these offspring will appear tall physically? One person should unmute and tell me.
3. 3.
3 of the
Thank you.
Three.
Tall.
Three,
One
ma.
is completely
Three,
tall.
ma.
Thank you. Two, they look tall, but they have a recessive gene for shortness, but unfortunately, one is very short. So a completely tall organism here is just 25%. We have 4, that is 100%. So 1 is 25. What about those that are tall but have shortness or short recessive gene? How many percent will that be? Can somebody unmute and tell me?
50%. 50%.
Thank you very much. Thank you. The short one also will be what? 25%. I hope you understand. This is just what it is. So if you are given any character, you can, if you find yourself working in an hospital or where you carry out genetic engineering, you can crossbreed. If you find yourself in the Greek region or employment later, and you are responsible to carry out crossbreeding, you have a very big orange, but the orange is not sweet. You have another orange that is very sweet, but it is small. You can crossbreed to have the character you want. I want a big orange that is very sweet. They have crossbreeded many plants like that. We used to have only white beans. Now we have the brown one. You have Oloyin, you have the drum. This was how they carried out many. Now you can have pineapples that are very, very short, but very sweet. Some are yellow, some are white, but sweet. Cucumber, everything, tomato, everything has been crossbreeded to have the character that we desire in our animals, in our plants. You can also do the same thing for microorganisms. All right, thank you. I'm happy that you all understand what we are doing. So now we are done with law of dominance, very easy. Law of segregation. Then we also have the law of independent assortment. This is a bit higher than your level, but I will explain because some of you will easily, even without teaching you, you will understand. So the law of the independent assortment states that inheritance of one pair of factors, that is the gene, is independent of the inheritance of the other pair. Fine, we've said that before. Alleles for different traits are described to sex cells or as sex cells and offspring independently of one another. Law of independent assortment emphasizes that there are separate genes for separate traits, which is true. The gene for tallness is different from the gene for shortness. The gene for color of the eye, white, is different from color of the eye that is gray. That is what he's trying to say after his study. And the characters sort themselves independently of the others. That is, that I have a brown eye does not mean I must be short. Because the gene for shortness is different from the gene for my eye. They are two different things. That is what the law of independent assortment is trying to tell us. That at the time of gamete or zygote formation, the genes are independently passed on from parents to the offspring. So you can see an example of organism, for example. Maybe the father is tall and is dark. The mother is short but fair in complexion. You now have the child. He's tall but is fair in complexion. Taking the tallness of the father, the fair in complexion of the mother, because these genes are separate things, they do not mix, so they can pick whatever. Anyone can be donated. So it's also the same thing with microorganisms. So during his work, Mendel discovered that the height of the plants, the shape of the seeds of the plant, and the color of the pod does not impact each other, like I have explained. The fact that a plant is tall does not mean that the plant must have a green pod. Neither must the green pod be filled with wrinkled seed. It doesn't matter that the plant is short, does not mean the seed must be wrinkled or bad. So the various features appear to be inherited independently. It is independent. It does not depend on each other. So that is what it is trying to explain here. So here you will see that if you, excuse me. So that is what the law of independent assortment is saying. So in a question involving two different traits, your answer will be independent assortment, 9 times out of 10. So this can be illustrated using a Punnett square in what is known as a dihybrid cross, meaning that the parents are hybrid for 2 different traits. The genotypes of the parent pea plants are illustrated. So we've been dealing with, for the law of dominance and the law of segregation, we've been dealing with only one trait, either tall or short, either red or white. But for the independent assortment, you are going to deal with different traits. So we want to look at two traits. We are looking at color yellow or color red. I mean, we are looking at color yellow and the shape of the organism, round. So this parent generation, we have a round, RR, completely round and completely yellow seed. It's going to mate with a completely green, which will represent small y, completely green, but wrinkled, that is small RR. The seed is wrinkled. For their first generation, what do you have? If you use Punnett square, just crossbreed them this way. You have the capital Y and the small y here, so you have YY. Then the red, you know you are using 2 characters now, the color and the shape. So for the color yellow, 1 capital Y, and then the recessive yellow in this one. That is 1 capital Y, and then Y, the small y. Then for the shape, this one is round, completely round. So pick 1, capital round, and then the recessive round, the small r. So for the first generation, this is what you have. You have an organism whose seed is what? What is going to be the appearance of this seed? Can one person unmute? Just one person.
The seed will be round and the color yellow.
Thank you. This seed for the first generation, that is F1 generation, the phenotype, it will appear yellow and the seed will appear round. But mind you, it has recessive gene for green, and then recessive gene for wrinkle. So what will happen now is for you now to segregate this now, and then you crossbreed the way we've been doing. That is all. And when you are done, you see that you have ratio 9 of completely yellow, ratio 3 of completely green, ratio 3 of wrinkle, and yellow, then ratio 1 of completely wrinkle and green. So that is it. So, but there are some problems with Mendel's laws. Even though Mendel's principle, his laws, form the basis of genetics till today, but there are some things that have been disputed. Some traits do not follow his pattern. He's still the father of genetics. But over the years, they discovered that there are some little disagreements with his law. Some things may not follow, some things may change. For example, incomplete dominance. They discover now that when you blend color, for example, it is not necessary that it must be red, because mixture of red and white can become pink. And that has been discovered with the gene. They also discovered the issue of codominance, that both alleles express equally. For example, blood type AB in humans, they are 50-50. So one is not dominating the other. They also saw the issue with multiple alleles. That is more than 2 alleles for a gene, e.g. blood type inheritance. Not that it must be, only one must be donated. So in a family where SS marries, they marries an AS, they may have all AS. They may also have all SS. So there are probabilities. They also discovered the issue of linked genes. That genes located close together on the same chromosomes do not as such, they may not separate, despite the fact that you try to separate them. So these are the anomalies or exceptions to Mendelian genetics. So in what ways have scientists been applying the Mendelian genetics? Mendelian's laws are foundational to genetics, like I've said earlier, and have been applied in medical genetics, that is, in the understanding of hereditary diseases, such as sickle cell anemia and cystic fibrosis. There are many diseases that medical genetics has helped to unravel. In those days, they will say, children with sickle cell, they will say it's the mother-in-law that is killing the children. It is one witch in the village. But later it was discovered that it was sickle cell, no more that the child is Ogbanje. So it has helped, the Mendelian genetics has helped to unravel several diseases. So you can avoid what you don't want. They will advise, oh, you are in love, right? But find out if your fiancé is an SS. If the fiancé is an SS, if you actually love each other, you will separate. You go and look for another person. I will look for, because if love blinds your eye, SS children will open it. By the time you start going to the hospital every day, you won't even have love again. There will not be time for love. So there are some other sickness that you know exist in some families through this genetics, and they can be avoided. In modern day science, in the advanced world, when a woman is pregnant and they take the blood sample of the baby, they can determine whether this baby is good or not, whether it has one problem or the other. So they can abort the baby if they know that it's going to be a problem. And that is where abortion laws is now raging in the US. It is also helpful in agriculture. I've told you, breeding of high yield and disease-resistant crops. Is it plantain? Is it orange? Is it beans? Our chicken, our pigs, everything they have crossbreeded. It's also helping in the forensic science, DNA profiling and inheritance tracking. See, when they kill somebody in the US, in the UK, anywhere, by taking sample in the body of that person and isolating some organisms, they can know which country the killer comes from. They can see, oh, they traced a particular bacillus, and the bacillus is from the tropical region of the world, and they can trace it to Nigeria or trace it to anywhere. That is forensic. You can use microorganisms to trace who kills a particular person or who rapes a particular woman. In microbiology, it can be used to study microbial genetics, antibiotic resistance, which is very important, and hereditary traits in bacteria. So Thank you very much. And I will conclude by saying that microbial variation and heredity are fundamental to understanding microbial evolution, adaptation, and practical application in science and medicine. Through genetic and phenotypic variations, microbes continuously evolve, influencing health, industry, and the environment. Like we know, some microorganisms are now used to treat diseases. For example, they can put some drugs and use maybe E. coli or non-invasive organism to drop the drug to kill cancer cell. Once they detect the part of the gene that is the cell that is affected, they can release that drug straight into that cell instead of killing other cells around that cancer cell. So microorganisms can be used to drop the chemical or the medicine in the right cells. And we know that through this genetics, they've been able to detect which organism can give us which enzyme and which enzyme can produce what in the industry, whether food, chemical, or any industry, and in the environment. Through this knowledge, you can isolate enzymes and use it to clean the environment. So Mendel's discoveries has revolutionized biology and paved the way for modern genetics. His laws of segregation, independent assortment, and dominance remain very fundamental a concept in understanding inheritance pattern in both simple and complex organisms. Simple organisms like bacteria, complex organisms like plants and animals, including human beings. Despite some exceptions in his principles continue to guide genetic research and applications in various scientific fields. Thank you very much, at least to some extent, but for a few 2 or 3 people, we had a very peaceful and interesting class today. And I'm happy because in the course of asking questions, I was able to detect that a very majority of you were actually following what I was saying. At least you have the confidence in crossbreeding and being able to use the Punnett square today. Thank you very much. So you can unmute and then I can take some questions if you have one.
Thank you, Ma. Please, I have a question.
Sarah Ayorinde is raising hand. Go ahead.
Good afternoon, Ma. I was going to ask, in most of the cases we have where parents with maybe tallness as dominant and shortness as recessive, whereby in those cases where we have the parent is short but they have a recessive tallness, how do we represent it in letter? Can we make the small t come first and the tall t at the back?
It doesn't matter the position. Even if you do that, when you crossbreed it, you will still get your answer. It's not the position that matters. If you turn it the other way around, so if you crossbreed, you should get the same answer. But it is only logical that something that is dominating should come first, isn't it?
Yes, Ma.
You cannot say that something is dominating, then you put it last. So don't see it as just letters. Letters are being used just for you to understand it because you cannot see those things with your eye, except you study them under the microscope.
Okay, Ma. Ma, my second question is,
okay,
is it really proven that some characters like behaviors can be inherited from parents?
Yes.
Now, apart from type and genotype,
that is what
character.
is called, okay, so I didn't know what point you came in. We talk about inherited, inheritable, heritable, and non-heritable. So character is part of heritable.
Okay,
so why inheritable?
Right.
What something you can inherit is genetic. You don't have a fault. It can be carried from your parent to your offspring because that one has to do with your gene. But for the heritable, it can have to do with character, but it does not necessarily mean it must happen. If you see that your parent is have this attitude and you don't like it, you can work at not being that person.
Okay, ma.
All right. So now then you have the non-heritable, which is at the phenotypic characters. Yes. And wow. But remember that we are not talking about human being in this course. Don't forget that we are talking about microorganisms. But we cannot talk about microorganisms without using human being to explain for you to. You get it?
Yes, ma.
Wow.
Yes. Good afternoon.
Thank you.
Yeah, I want to know if there's any case that has been recorded whereby two dominant SS parents crossbreed and give rise to a dominant AA.
No, it's you cross it yourself. Cross it yourself. Cross it yourself. It won't
I
happen.
want to, yeah, I want to know if there can be a mutation during crossing.
Mutation.
Yeah.
Well, I won't say yes, because I know miracles does happen. I've heard about that. I've heard about SS turned AS. And let me just tell you too that they just said that there are some exceptions to Mendel's law. But SS and SS, nobody is going to tell you to do that.
Okay. Thank
Wow,
you.
you put down your hand so that I can see the other person that is raising. Olanike Atolagbe. Yes.
Oma, you just answered my question
Oh,
because I wanted to
thank
ask about the
you.
into AA that
Please, you guys that you
Thank
are asking me this question, if you know you have a girlfriend that is SS and you are SS, or don't even joke with it, please just, if you actually love that person, the best thing is for, if you are AS and AS, the best thing is for you to just separate. Because yes, you may have children, you may be lucky to have two children that both of them will be AS, but what if if the person is unlucky? We've seen a family that have two SS and one AS. Do you know what it means to take care of two SS children? I would do praise, Ayowa.
Good morning, ma. Sorry, ma. I had a network issue when you were explaining the law of independent assortment. At least I stopped at where you started explaining with the Punnett square.
Okay, so you will continue by listening to the video all over. You can do it on YouTube. I can also send the video, the YouTube link to our platform. I will not be able to go over it again.
Okay, ma.
Thank you.
Thank you.
Any other observation, addition? Real one, are you in this class today? Oga, real one.
Please, will you send the slide?
Oh, I've been sending slides. Is there any slide I did not send to you? Please, if you think there's any slide you don't have, you better speak up. Your class captains are on the chat group, so they should have sent you your slides and your video. If they don't, then let me know. But again, you are registered on the university LMS, the Learning Management System. I also take the pain to go there to upload your video link and your slide. On the platform, you have it. For last week's lecture, you also have the YouTube link. And for today, I've already sent the YouTube link. I will resend it again. So if you have any observation, any other thing, please let me know. Shodipo, you are asking, how do you get the link? Please ask your class captain, because I will copy the link and put it on the class captain platform. It is expected that the class captains should give it to their class member. This person asking the question, Shodipo, I don't know which department you are from. So ask your leader, your course leader for each department, where is the link? Where is the video recording? The LMS link, there's no LMS link. LMS link, you have to log into the university platform. I log in there and I drop for you. You are registered there. All of you are registered. So if you don't know how to use the LMS, too bad. But I know some of you will not know how to use the LMS. And that is why I take the pain to also drop everything immediately now. So I need the time. That is why I need, because you give me extra duty to do, because it will take me another 40 minutes to be able to do all this, to send all these things to you again. So in the absence of any other question, can we call it a day, please? Any problem? Angela Kumilayo Fafiolu, are you okay? Abraham Ogenetega, are you fine? All right, so in the absence of no question, let's call it a day. Have a lovely day. Thank you.
Thank you, ma.
You're welcome.
Thank you, ma.
All right.
Thank you, ma.
Bye. Bye. Bye.
Thank you very much, ma.
God bless you.
Thank you, ma.
Thank you.
God bless you, too, ma.
Yeah, thank you. Amen.
Thank you, ma.
Thank God.